then is, oh, thank you. What I would like to do then is, firstly, thank you all for coming. I really appreciate you attending this pre-AIM webinar for Tomocube today. We will be at AIM next week with a system available for use as needed. So if you are interested, please feel free to put a note in the chat and I can get back to you personally to arrange that. I also want to thank the organizers of AIM as well, Feather and Holly, and the rest of the team for organizing the event. So with that in mind, I want to introduce Paul, who is our speaker today. Paul is a professor at KAIST in Korea and is the founder and CEO of Tomocube. So he will be walking us through some of the features and the kind of background of the technology of holotomography. If you have any questions, what we're going to do is about a 40-minute, 45-minute presentation, and then 10 to 15 minutes of questions. So feel free to put questions in the chat. I will make sure that they're answered at the end. And apart from that, thank you, Paul, and please go ahead.
Okay, good morning. Thank you for joining this webinar, and I'd like to talk about the holotomography from the principles and the benefits of using holotomography and some examples that we can utilize holotomography, some various, the typical study of the bioscience. So to make a long story short, I'd like to share what it can provide using HET. So it can provide three-dimensional high-resolution imaging of live cells and tissues and organoids without using any labeling agent. So no fixation, no staining, no fluorescent proteins, but you can see this like subcellular dynamics. For example, you're watching mitosis of the cancer cell line, chromosome condensation and separation, and those all this jiggling like the spots are corresponding to lipid droplet. You can beautifully see the membranes and also the major subcellular organelles. So I'd like to talk about starting with the quick summary of the history of live cell imaging. So from the development of the label-free techniques such as spatial controls for DIC, which is still widely used in the many research areas, that can open up the doors to the cytologies and cellular the dynamics. And after the invention and development of the various fluorescent labeling techniques such as fluorescent proteins and confocal microscope and super resolution, and now we have access to the molecular specific information, which is great for the study of the molecular cellular biologies. But now we see the areas of the 3D biology, such as the microphysical systems and organ-on-a-chip and organoids, and regenerative medicine like embryogenesis, stem cell biology, and therapeutic application. So we believe 3D high resolution, the label-free is very important for the future application and those in the topics. In the context of 2D imaging versus 3D imaging, if you use conventional microscope, such as the fluorescent confocal microscope, you have to prepare the cell to express fluorescent protein because the biological cells are transparent without using exogenous labeling agent because they are composed of the mostly water, protein, nucleic acid, and lipid, and they are all transparent in visible wavelengths. So you have to spend like hours to days of preparation in order to express the fluorescent proteins. And if you use conventional fluorescent microscope, you can get beautiful fluorescent imaging with molecular specificity, but you have to discard the cell because of the and after measurement, because of photobleaching and phototoxicity issues. You cannot use those fluorescently labeled cell for therapeutic application for obvious lesions. And most of the imaging techniques are optimized for the 2D layers of the cell for imaging. If you want to image like 3D samples such as sick tissues and organoids, you have to inevitably section this sample into thin slices, followed by staining and imaging. So which prevent from long-term imaging of the cell. But if you use HET, it does not require any preparation, no fixation, no staining, no fluorescent protein. You can just move your multi-well plates from incubators to our microscope, or you can utilize the tabletop incubators has already been installed in our microscope. So you can just keep watching and imaging the cells over days and months. We can also measure 6 samples up to 150 micrometer in thickness. So you can just keep watching the organoids growing over time, and you can also watch the stem cell colonies developing over the days and the weeks. Let me briefly explain the principle of HET. So HET is an optical analog to X-ray CT, computed tomography system. As you may know, the CT measures multiple 2D projection X-ray images as a function of various illumination angles. From those multiple 2D stacks of 2D imaging data set, you can reconstruct 3-dimensional tomograms of a human body without having invasive process in terms of X-ray absorptivity. And we can do the same thing with light, and we measure, we control the illumination angle or the illumination patterns, and then we are measuring multiple 2D holograms of the unlabeled cell as a function of illumination patterns or the angles, from which you can reconstruct 3-dimensional distributions of the refractive index. So refractive index is a physical quantity that describes the speed of light as the light is passing through specific materials compared to that of the light in vacuum. But the biological meaning of refractive index is protein concentration. I'll talk about the detail in the following slides, but I have to emphasize that the refractive index is intrinsic optical contrast, so that by exploiting this parameter for imaging, we can achieve label-free and high-resolution reconstruction of a cell. So even though this X-ray CT and the optical HET seems like a different technique, but they share the same physical principle, which is inverse of wave propagation. So compared to the conventional optical microscope techniques, such as if you use phase controls for DIC, it can provide label-free imaging, which is great for long-term and live cell imaging, but it only provides 2D qualitative imaging set. And if you use fluorescent microscope, it has the powerful molecular specificity. So you have the information where the specific proteins or the molecules are located in the inside of the cell. But you have to modify the cellular the state because of the use of exogenous labeling agent, and you have inevitably have the issues of photobleaching or phototoxicity. Compared to those techniques, we have no issues of the photobleaching and phototoxicity, but you can have structures of the major subcellular organelles. So this is the HAP3B cancer cell line, and we have the clear views of the cellular structures, such as the cell membrane and nucleus membrane and nucleoli. Those are mitochondria, and you can also see the lipid pyramid here. Our resolution goes down to 150 nanometers, and we measure 3-dimensional volumetric data, but I only provide the display one of the middle section for visualization purpose, but we have the stacks of data in tomographic reconstruction. And those are corresponding, those are not artifacts, those are the extravascular structures. So our imaging has high contrast, so you can detect the smaller object over there. So the use of HET has several benefits. So since we do not use any exogenous labeling agent, no labeling is required, so you can just readily measure the cellular and tissue imaging without any time delay and without any modification. And since we do not use any labeling agent, there's no issues of photobleaching or phototoxicity. You can just measure for forever as long as cell maintain their physiological condition. We can also achieve high speed and long-term imaging. So our speed is 100 times faster than confocal microscope. For example, our second generation, the system can measure the one field of view is like the size of 200 micrometer by 150 micrometer by 50 micrometer. So it takes only 5 seconds to measure the one tomogram. And then since it does not require any labeling agent and no photo bleaching or no phototoxicity, you can just measure for forever as long as the cells has maintain their condition. So we have also the stage top incubators in our microscope, so you can just keep culturing the cell over the many days and months. And our resolution is very high, go down to 150 nanometer, and we can also measure the leadless three-dimensional tomogram. And we also provide quantitative imaging information, but I'll talk about the detail in the following slides. So this is one of the screenshots that we can measure for the monolayer, the cancer cell line. So one slice images of our tomogram, and where we can see the various subcellular structural information, such as lipid drumlet, and nucleus membrane, and nucleoli, and mitochondria, and so on. And compared to conventional microscope, for example, in this case, we have used the fluorescent proteins to specifically label the mitochondria. And we used the 3D fluorescent microscope, which is also integrated in our microscope system. So in this case, you can nicely see the presence and locations of mitochondria in the cell in the middle. And if you use the HT mode, you can see the well-matched imagings captured by the holotomogram. But you can also notice that there are other adjacent cells which are expressing the mitochondria, but those are not picked up in the conventional microscope because of the many lesions, like the low expression levels and the low imaging contrast and so on. But we do not miss any of those information in HT mode. So due to the label-free features, we have no issues of the photo bleaching and phototoxicity, so we can enable various subtle investigations, such as in this case, we are watching primary culture neuron cell in 3D tissues layers. Obviously, this is in vitro measurement, though, but you can see the transportation between neurons and also the dendroid structures over there. And if you watch the cell in the bottom left, they are trying to connect by moving, and the changing is the lamellicolia, the cone shape. So you can just measure the long period of dynamics with a very high spatial resolution and temporal resolution. And in this case, you're watching HAP3B cell line, and you can nicely see the mitochondrial dynamics, and fission and fusion can be easily picked up. And you can also see their interaction with other subcellular organelles, such as the lipid drumlet, and you can clearly see the nucleus membrane here, and other major organs or subcellular organelles can also be easily seen, and also membrane ruffling can be picked seen from the movie. So our system has incubator inside, and you can easily combine with other perfusion system and microfluidic systems. So in this case, we demonstrate a simple experiment where we have placed the HAP3B, and first we perfuse the normal cell culture medium, and we just change it into the H2O2. So now you see how cells are dying. Mitochondria are being fragmented, and you can also see a few cells has gone and the formations of vacuoles. And when you perfuse to the normal cell culture medium, not all of them, but a few of them are going back to the healthy condition. So they are just reviving. So you can see the recovery of the mitochondrial shape and removals of the vacuole structure, which is not easily labeled with conventional methods. So this movie is taken for only 2 hours. So it is changing very fast and dramatically in biological stage. But if you use conventional method, this dynamics is too fast to be captured by the conventional methods. And we can also measure for a long period of time. So this is the 1 week's movie. So one of our users in Switzerland, they were combined this system, HET, with the specially designed microfluidic device in order to capture the individual yeast cell and track how they are undergoing the the multiple round of fissions, and in order to study the cell aging over the 1 week of the time frame. So without having any issues of the vibration and stability, you can just nicely track those dynamics with high resolution by using this, microfluidic device and for fusion for the investigations of the structural information of over a few days and up to week. And when it comes to the long-term imaging, if you use conventional fluorescence-based microscope, usually you may be able to see like the hours or days of movie with very limited and very specially controlled the environment condition. But when it comes to the holotomogram, long-term imaging, we can just measure for weeks and months. So in this case, we have started with the patient-driven fibroblast and differentiated into the adipocytes over 1 month and a half, over 42 days. So I have to say that this is exactly same field of view captured using our microscope. So you can see the differentiation into adipocytes and the formations of the clusters of lipid drumlet inside of cells, and the cells are becoming like the multi-layer tissue-like structures, and also growth of the individual lipid drumlet over here. Now we can also measure the larger field of view. So this is the movie taken at the days of 42. And even though our field of view is about like 200 micrometer by 150 micrometer by 50 micrometer volume size, but by measuring the multiple field of view and stitching them, which is provided by our software, you can just measure a millimeter by millimeter size in tile scan. So this is 4 millimeter by 4 millimeter in 3D, and where you can see the seamless, the stitching data, and you can see the the individual lipid drumlet as well as the subcellular structures. Like Google Map, you can just zoom in and zoom out using our software. Okay, so as I said, the label-free imaging has the some benefits in measuring the long-term dynamics. So in this case, we have compared our HT data in comparison with the conventional fluorescent dye, such as lipid dye, in order to label the lipid drumlet here. So after 30 minutes, due to photobleaching, signals goes away, and this is irreversible, as you know. But using HET, you can just keep monitoring the cellular structures without having any issues of photobleaching. We can also measure the three-dimensional, the sick tissues, sick samples. Can you guess what is this sample is? So this is a embryo, the mouse embryo. So size of this embryo is about 150 micrometer, but we can measure the samples to the size of the 150 micrometer. So it takes like the in a few seconds to measure one tomogram. You can nicely see the two cell stages and also the nucleus structures here, the cellular structures here, and you can also surrounding the sperm cells in the mouse sperm cell. So combining this three-dimensional volumetric imaging capability as well as the label-free imaging, so a few of our users are applying this system for the study of the embryogenesis. So you're watching the same embryos from days of from the two cell stages and up to the blastocyst stages, and you can also have the high subcellular imaging capability in order to detect the nucleus membrane and nuclear light and other subcellular structures. So they are studying the mouse embryos, but some of them are interested in moving toward the human embryo studies. So they are studying in order to get the certificate for ILB in order to move on to the human embryo. I think that will be very interesting to see human embryos, the data without having any photobleaching and phototoxicity issues. Stem cell biology is also one of the very important applications of holotomography. compared to the bright field microscope, which only provides the qualitative, outline structures of the clusters of some say colonies, but we have high contrast and volumetric imaging capability, so you can see the individual cell in multiple layers with their subcellular imaging capability. So other types of three-dimensional samples, such as organoids, can also be measured with subcellular resolution. So this is live organoids, and like the shell-shaped structures, and diameter is about 150 micrometer. And for visualization purpose, we only show the bottom half, and you can see subcellular structures like that. And important thing is, we didn't change any protocols. So organoids is happily grown in the matrigels, and no clearing, no refractive index matchings, and no additive, no protocols is changing. So organoids is happily growing in this condition, and we can have their various dynamics like the mitosis and apoptosis and the structures of the subcellular organelles. We can also measure the small intestinal organoids from mouse, and it starts from the size of 50 micrometer. And this is another 1 week's movie. So we have incubated these organoids in the tabletop, the incubators, which is incorporated in our microscope. And this finally organoids goes to the size of 150 to 200 micrometer. So you can see the individual cell and their subcellular structures. But we have several limitations as well. So currently, our penetration depth is limited up to 150 micrometer. So which means your sample is greater than or thicker than 150 micrometer, you may have some blurring effect like that. So this is one of the limitations that we have to use current commercial system. Nonetheless, the small organoids smaller than 150 micrometer in thickness can be measured without changing any protocols, or without any sectioning, nor the refractive index matchings, nor any clearing. Okay, so other types of three-dimensional tissues, like the volumetric histopathology samples, can also be measured. So in this case, this is the fixed mouse brain, but fixation is it's not mandatory. This is optional. We can also measure fleshy tissues with the high resolution and high contrast. So conventionally, sample is prepared with a thin slice with a thickness of like 3 micrometers or 5 micrometers, and followed by the staining and seen by the bright field microscope. But in this case, we have prepared the samples to have the 150 micrometers, so which is about 50 times thicker than the conventional slice. But without staining and without thin slicing, you can see the individual cell. Like in this case, you are watching the dendrite structures and the nucleus and nucleoli and multiple layers, because this is volumetric 3D data set. Okay, so some of the users and collaborators are trying to find some the applications we're using HCT. So in this case, they are interested in developing the drug response to the organoids for the alternative testing and drug discoveries, and for various applications. So they were studying the growth of the small intestinal organoids in the response to the the target drugs targeting the cancers therapies, and they were observing the individual cells such as goblet cells and penetrant cells, and also observed mitosis and apoptosis, and have tracked the volume and the mass of the individual cells and the total the organoids. And one of the benefits of using HCT is quantitative imaging capability. So I have mentioned that the principle of HCT is the reconstructions of three-dimensional distribution of refractive index. And refractive index is a physical quantity that describes the speed of light at the specific positions of the sample as the light is propagating in the middle of the sample. But its biological meaning is the biological concentration. There is the linear relationship between the reflective index value and biomolecular concentration, which is known from 1950s and has been well documented and studied in the biochemistry. But there was no technique. Technical development was limited in order to visualize the three-dimensional distribution of the live cell with high resolution. And now we have technique and commercial device, so you can measure three-dimensional distribution of reflective index in quantitative numbers. And this is physical quantity, so this is highly reproducible and highly quantitative numbers. So, for example, this is one of the slides of tomogram or reflective index of hepatocytes. And those, spherical structures are corresponding to lipid drumlet. And if you leave the value of reflective index, for example, this point has an index of 1.38, something like that. And from the prior information, which is documented in biochemistry, this slope, you can retrieve or calculate the concentration of lipids in the lipid drumlet in the units of gram per liters. And also we have volumetric imaging capability. So you have the volume information of the individual, the lipid drumlet in femtoliter units. So you have two quantities, right? Concentration in gram per liter units and volume in the liter units. If you multiply those two values, you can calculate the dry mass of the individual lipid drumlet. You can also apply to other subcellular organelles. So you can measure mass of the nucleoli if you want. You can just measure dry mass of the bacteria of the cell without sacrificing this individual cell. You can just measure and monitor. So this is not well appreciated in the field, but some of the users have recognized the the potential of this feature, so they are applying to the various applications, including the phage separation and the LLPS, lipid in the phage separations, and also the bioengineering, where some companies are using bacteria or algae in order to produce the high valuable products such as bioplastic, PHA, something like that. So they were using HET to quantify for various applications. Okay, so in order to facilitate this analysis, we also provide automatic software. So unlike the other subcellular organelles, lipid drumlet has distinctly high reflective index value compared to other subcellular organelles. So by using simple stress holding approaches, you can precisely segment out the lipid drumlet, and we provide the software to conduct this analysis in 3D. So the measurement was done in 3D, and analysis can be done in 3D. So you can calculate and follow the growth of the individual drumlet, and you can retrieve the quantitative values about volume, concentration, and dry mass of the individual lipid drumlet in various applications. Okay, so in this slide, I'd like to compare some features of the microscopic imaging for the cell analysis. So this is called the pyramid of frustration proposed by the Eric Bachik in his papers. And it was said that the several features are important for the study of the cell analysis, including spatial resolution, temporal resolution, signal-to-noise ratio, and sample size are all important. But each technique has different pros and cons. So for example, if you look at the confocal microscope, their SNR is great because of the use of the fluorescent protein. But if you speed and sample size and temporal resolution is somewhat limited. If you use super resolution microscope, the resolution go down to the tens of nanometers or even like the nanometer scales in some cases. So sample spatial resolution is great, but that is done by sacrificing the sample size and temporal resolution. Light sheet microscope is great technique. It has the low the phototoxicity and very high speed of acquisition, but still it uses the the markers. So sample health is still not good. So HET can provide the best sample health condition because we didn't change anything in terms of the sample preparation. No staining, no exogenous labeling agent is required. We can measure over a long period of time. Our spatial resolution is slightly better than confocal microscope, but worse than super resolution. And our temporal resolution is 100 times better than the confocal microscope, and comparable to the light sheet was slightly faster than confocal microscope. Our signal-to-noise ratio is good in terms of identifying the major subcellular organelles, but we have some limitation. We do not provide molecular specificity, but this is different data. So this is the summary of the specification of our second generation of HET. The reason why I mentioned second generation of HET is because there was a first generation of HET based on laser, and the first generation only measured thin slice. But when it comes to the second generation, it used LED, and which means there's like sample viability is much better than using high power laser. And also, it does not have any speckle noise, which was induced by the use of the laser. And it also has 4-channel fluorescent for 3-dimensional fluorescent imaging based on the convolution algorithm. And we can overlay 3 channels, and we have the incubators inside, and we can just measure long period of time. So since we have both HET and 3D fluorescent, the features provided the same machine, you can perform correlative imaging like PET-CT. PET provides when PET-CT was used together, the CT provides structural information and PET provides the molecular specific information. We can do the similar thing. The HT can provide structural information of the cell, and the 3D fluorescent can provide the molecular specific information. So in this case, the RP and miter tracker and the lipid dyes are overlaid on top of the HT data. So you can just compare, you can just generate, investigate various in the application by using these 2 different data sets in 2D cell layers and also 3D organoids in a 3D culture system. Okay, sorry for using this Korean, but I like to emphasize that the software is very easy to use, and you can organize your project, and in each project the data can be organized using the software, and we also provide cell segmentation features without using the Python code or the open source external features. Everything can be done by using the integrated software. So by clicking a few buttons, you can perform cell segmentation or subcellular organelle segmentation. I guess we can also provide the on-site the demonstration after my presentation. So we are now also developing the various advanced cell imaging features, which is not commercially available yet, but we have platform to integrate the newly developed the AI-based cell analysis, which will be followed in a few months frame. So I also talk about some ongoing developments in the context of the AI analysis. So I'll just skip this part. And so HT is a relatively new technique in the field of biology and clinical research application, but leading institute has already developed introduced this system, and they has generated various interesting study in the field of the phage separation in biology and microbiologies and cell biologies and so on. So I do not have, for the sake of time, I cannot go to all the details of their publication, but if you're interested, you can just refer to our webpage. You can also see some papers generated by using the HT system. Okay, so far I have talked about the basic principle of HET and benefits of using HET with some representative imaging result. From now on, I like to talk about ongoing research project where you can see that how HT can be combined with AI for various applications. I have to note that this is not commercially available as of now, but we are working on the several project, and we are in the process of translating this AI-based research result into the integrated software so that our users can readily use this AI-based more advanced analysis of cell the features. So I cannot talk about all the details, but I will just talk about some representative approaches in the combination with the HT and AI. For further information, you may want to refer to the review articles published in Nature Medicine from months ago. So when you combine the label-free imaging technique, such as HT, with AI, various approaches can be achieved. Research can be achieved. For example, you can improve the imaging qualities. But more importantly, you can just perform cell segmentation or the subcellular segmentation using AI. And you can also do cell type classification. And you can also virtually generate the artificial fluorescent data or the molecular specific information from the unlabeled cell, which is called the virtual staining. So I'll talk about the representative study in the following slides. So in this case, you're watching the CAR T cell in the back, which is the interacting and the killing the target leukemic B cell in the front. And this is the 5 hours of movie. So after forming immunological synapse, this CAR T cell is killing this target cell by inducing apoptosis. So you can see the membrane blebbing here, a hallmark of apoptosis, and this target cell is being killed. If you keep watching, you can observe that this CAR T cell is moving to another place, kill another one, and so on. So like you can just perform the potential assay if you want. So this is label-free movie in 3D high resolution, which is great to generate this kind of eye-catching movies. But important thing is what kind of information we can retrieve automatically and systematic manners. So to do that, we wanted to provide the software, the algorithm to perform the automatic segmentation. So this is the result of the applying AI for segmentation. So AI has automatically recognized which one is target CAR T cell and which one is the effector leukemic B cell. So again, this is label-free, so cells are very transparent and happy and alive. But using AI, we can virtually stain effector cell as the blue color and target cell as the purple color. So the power of AI is scalability. So you can apply this algorithm to the large field of view and time-lapse data. So that cannot be done by the manual segmentation. So we have further developed the algorithm for segmentation. So now we are able to perform segmentations of subcellular organelles from this one input data. So we are internally keep improving this algorithm so that it can be readily used by the users. Second categorizations of the application is classification. So you're watching a movie of the bacteria. So you start from the single Bacillus subtilis bacterium. And if you watch, you can see how bacterial cells grow and divide and grow and divide. And finally, they are forming the bacterial film-like structures. After watching these movies, we saw that we can see inside of the bacterial cytoplasm. So we thought it might be possible to utilize this information for the bacterial species classification. As you know, bacterial cells has a similar shape. So by using conventional bright field microscope, it is not possible to differentiate bacterial species. But we have information in 3D, in subcellular structures, and our hypothesis was that different bacterial species may have the different protein composition and structural distribution. So we may be able to train this algorithm by combining HT for the cell type classification. It's like finding catch a dog out of the one photograph. So in collaboration with the Samsung Medical School in South Korea, we have prepared a sample from 19 different bacterial species, which are frequently found in bacterial sepsis patients in South Korea. For each species, we have collected 300 to 500 the HT data set, which doesn't take too much time because our field of view is very large. And in one field of view, you can collect multiple bacterial cells, and you can also perform automatic segmentation. And after measuring those data, we have trained the convolutional neural networks for classification. And after training, we tested the algorithm with the single bacterium inner samples in order to see whether this algorithm can predict the species of this sample. So once we measure only single bacterium in a sample, accuracy was like 82%. But if you take 5 bacterium in the samples and measure and average, perform ensemble averages, accuracy goes above 99.9% here. But after that is the result published in 2 years ago, and after publication, we found that there was one mistake in the algorithm in order to optimize the algorithm. So this is now the current results. So even though you measure the single bacterium, accuracy is above the 95%. So we are doing some preclinical studies with the medical centers in South Korea to see whether it can be used for the clinical the settings. The similar approaches can also be applied to the bone marrow, white blood cells, and also the white blood cell found in the peripheral blood. So the 5 differential can be done easily, and TBNK can also be done. And we are also able to detect CD57 positive cell out of CD57 negative cell, which are related to the immune cell aging. So those are some ongoing projects. So we are trying to understand the potentials of the cell classification by using HT and AI. So we are investigating the several project in order to see what is the ultimate accuracies and what is the limitation and challenges. So this is the research level activities. So another applications of combining HT and AI is virtual staining. The reason is every technique has pros and cons. So HT also has potentials and limitations. One of the strengths of the HT is like label-free, 3D, high resolution, and fast imaging. But HT also has limitation. One of the key limitations of HT is the limited molecular specificity. What I mean by that, even though we can generate the high resolution 3D structural information of cell without staining, we do not have molecular specificity. So for example, in the lab HD data, if you ask where's the actin cytoskeleton, we have no idea because of the limited molecular specificity. So that's why many of our current users are actually measuring the fluorescently labeled cell using our microscope. So first they measure HT and followed by acquisitions of 3D fluorescence. So in their papers, they provide both HT and the multiple channel fluorescence, which is good for correlative study. But the criticism is that we are claiming label-free, but our users are still using the fluorescently labeled cell, which is correct, which is the status as of now. But we're trying to move forward so that we want to provide both structural information and the molecular specific information without using any staining agent. So I just skip this part. And we were trying to combine HT and AI. In particular, we are trying to apply the GAN networks to the HT data set for virtual staining. So what we have done, we have first prepared the fluorescently labeled cell, and using our microscope, we measure both HT and the multi-channel 3D fluorescence for the same cell for training. And after training, you don't have to label the cell anymore. And by applying the AI algorithm to unlabeled holotomogram data, algorithm can virtually generate 3D multi-channel fluorescence. So accuracy was very high. So as you can see here, the left column is the raw data for various different types of the cell line. And all the light columns are corresponding to the virtual the fluorescent data in 3D. And accuracy was greater than 97%. So you can perform this kind of a spatial biological study. So this is a movie of the cancer cell line measured by our imaging microscope. And the right columns are corresponding to the virtual staining data in 3D. So now we are moving to virtual staining of H&E to the volumetric tissue samples, and we're also working on the virtual staining of the IgE so that we can apply this virtual staining to the samples that you do not want to use the fluorescent labeling agent, such as the stem cell and the growth of the immune, the organoid cells and the neuron cells and sperm cells and embryo cells. So those are the projects that we are working on right now, which is not commercially available yet, but the aim is after peer review validation, publication, and we are trying to provide the software so that users can readily adapt the AI algorithm for applying this algorithm to their data set. Okay, so I like to conclude my talk with this slide, which I like the quote that I like. So Robert Oppenheimer, the physicist, once said, "The deep things in science are not found because they are useful. They are found because it was possible to find them." And it is our hope that we want to provide the best cell analysis solution so that the scientists like you can perform the new investigation in order to find the new finding. So with that, I'd like to thanks for your attention. Thank you.
Thank you so much, Paul. We have a couple of questions that I'd like to go over in the next. We have about 12 minutes left. So the first question was, when you image the same field over 42 days, does this mean the sample sits in the microscope for the whole time, or can we take it out and refine the same area by some means? So how was the 42-day time lapse achieved?
So I think the DJ has already replied, but in addition to the DJ's reply, I like to add that you can there are several options. So as the DJ has explained, so there's an options to slightly move the stages in order to change the medium and perform the additional measurement for timeless measurements, or you can just move back and forth between our imaging microscope and the external incubators. And since our mechanical stability is so high, you can just trace the positions of your interest. So the software can automatically move the position without any the issues of the localization.
Yeah, no, that's perfect. Another question that we had was, which subcellular structures and organelles can be visualized? Is it possible to image the ER or vesicles?
I think that is the great question. So I think let me go to the slides that I have used. Let me just go back to this one. So these questions is related to potential and limitations of ACT. So as I said, what we measure is the three-dimensional distribution of refractive index. So we can precisely measure the information of refractive index in 3D. So, but the thing is, the different part of the cell, like the different subcellular organelles, they have the similar the overlapping values of the refractive index. So it is not possible to differentiate the subcellular organelles only based on the refractive index value. This is also related to the our potential because of the different part of the organelles or different species of the proteins. They have the same slope, which means it is great to quantify the information because the slope is same for the many of the organelles and the proteins, but that is inevitably the prevent from providing molecular specific information. So this is why we are trying to, let me just erase this information. Nonetheless, as you can see, these movies, like these images, the major subcellular organelles can be detected from like the low data set. But as you have the mentioned in your questions, not all the subcellular organelles can be detected from this low holotomogram data. This is why we are working on the AI-based analysis in order to retrieve the segmentation, the virtual staining, the data from the low, the unlabeled cell information. So even though we haven't done the study for the ER or the other types of vesicles, but I think in principle it might be possible to pursue and investigate. I think that is the open question. So we have validated the major subcellular organelles can be virtually stained, and the next question is to how much we can differentiate, like the whether the ER can be visualized or the endosome can be visualized, and those are the all open question.
Yep. Next question is, can we visualize apoptosis in organoids?
Oh, okay. Great question. So
Yeah.
yes, we do. And I have to say that, let me show you the movie here, image here. I think that is the somewhere around here. Okay. So I think this is the the preprint where you can see the I think I'm not sure whether this is apoptosis or necrosis, but we can see we have detected the cell death in the organoids without any changing of protocols. So I have to say that HT is a the relatively new technique, and it has some potential, but we do have very limited the publication from ourselves and our users. So I think there are many like the potential opportunities to investigation. For example, so these questions of cellular apoptosis, we have detected on the some events here, but if you ask whether they can be automatically detected by using software, I think that's the way to go. So the users can develop like their own the algorithms, and some of our users are interested in developing the AI-based further the advanced analysis, like the detecting the apoptosis like that. So this is also the good topic of study, I believe.
And an extension to that was also, what type of quantitative information can we get from imaging organoids?
Okay, I think that is also a good question. So this is the very beginning of the study. So we have subcellular imaging capability of the live organoids without clearing, without sectioning, and without staining. So we can just measure days and weeks of the movie of the growth. I think this is the first observation that no other technique can provide similar features. So the next feature is what kind of information can be retrieved. So that's why many investigators are trying to use this machine for their studies. Like for example, our collaborators are interested in the how the dry mass of the individual cells are changing as a function of time in response to the various types of the drug candidates. For example, the collaborators are interested in applying this technique for the companion diagnosis. So they are interested in applying the patient-driven organoids in response to the potential drug treatment to see, and they want to see how many cells are alive and how many in the necrotic or the apoptotic cells are observed as a function of time. So those are all the research topics that you can pursue.
Can we do cell counting?
I think yes. I think this is very straightforward that we can also do cell counting, and that it can be done by using our commercially available software. I believe DJ can also do some demonstration in the following session.
Yeah, that would be something we'd be happy to show next week when we're at AIM. It's very achievable with our software. What is the minimum difference in refractive index that the Tomocube is able to detect?
Okay, I think this is very important questions about our features. So we can detect, we say the sensitivity. So how sensitive we can distinguish between like the index changes. So our sensitivity is like a 10 to the minus fourth, 0.0001. I think this is very sensitive to detect the small variations in space and time. So for example, I like to remind you this movie like that. So those are not artifacts. And as you can see here, the background is very flat, and you can even detect the heterogeneity in nuclear plasm. I'm not sure how good you can view this movie because this is the Zoom in the online, sharing. So because our resolution is too good, you have to use a Retina monitor to fully see our resolution. So I like to suggest that you may want to go to the YouTube channels of Tomocube, and when you view the movies, there's the gear box in the bottom right. You have to click and you have to check the 4K options to see the resolution.
Yeah, I know that DJ and I recently worked with a customer on expanded tissue, and we were able to fairly easily detect 1,000th of a point difference between certain elements in that. So it's very, very high resolution for refractive index. The next question is, we are implementing our own analysis at Tomocube, but can the user implement a deep learning custom macros to analyze the images? What is the format of acquired images? Some of this we can definitely follow up with offline. But Paul, would you mind talking a little bit to how we might work with customers to help them implement their own deep learning?
Yeah, I think this is a great question, and I totally agree on the point. So we already has some features to export our data to other format like the ImageJ and the cell pulse analysis. So you can just export our data, Holotomography data to other the cell analysis format or the Python or MATLAB or C++. So some of the advanced users who are familiar with the deep learning analysis, like this deep learning cell analysis in specialists, they are already using our data set by exporting to their platform. But we realize majority of cells biologists are not familiar with the this kind of the advanced analysis, and they are not also even familiar with the open source. Like some of the users have the trouble in fully using ImageJ and other software. So that's why we are trying to make a simple analysis software so that they can save their time. So we are doing the two approaches simultaneously. Advanced users can readily use our data set for their analysis, and we are also providing the software internally developed so that the scientists who are not familiar with this kind of advanced imaging system can still use the cell advanced analysis, the features.
Yeah, so I realize that it's now 11, so please feel free to jump off. We will continue for a couple of minutes answering questions if people are interested. can we acquire high quality images of C. elegans tissue and cell morphology in vivo?
Quick answer is yes.
Yeah.
We can see even bigger samples. I think DJ will show you some of the demonstration movies.
Sure. Sorry, next question. How sorry, one sec. I missed that question. How does the Tomocube deal with similar structures, e.g. an organelle that has differing refractive index in different parts of the cell due to differential aggregation of proteins, viscosity, etc.?
Okay, I think that is this question is related to the previous question. So as let me briefly repeat my previous and the response. So using so we can provide the precise reflective index reconstruction, but the value of reflective index is not good enough to distinguish the subcellular structures. So we can provide measure the subcellular structures such as nucleus membrane and the nucleoli, mitochondria, lipid drumlet, and so on. But the small subcellular organelles and other subcellular organelles cannot be readily distinguished from our raw data. So that's why more advanced imaging features, such as by combining AI, it might be helpful. So this is the ongoing project we are trying to achieve. But for example, we can also do another similar alternative approaches. For example, if you are interested in studying like the endosome, for example, but if you use conventional methods, you have to prepare multi-channel fluorescence, like the membrane markers and endosome markers in the nucleus and mitochondria, for example. But since HT can readily provide the major subcellular organelles without staining, you can use single fluorescent channel for endosome, for example. So I think that is practically important for many of the current research of the project, I believe.
Yeah, I think that's a case where doing correlative imaging makes a lot of sense with the HT and the fluorescence to achieve an even better result. One more question about the segmentation methods being developed. Can these also be applied to phase contrast X-ray micro CT images of tissue as a single channel image data is of a similar type?
I think in principle it may work, even though we haven't tried. But since we are developing the segmentation methods, which is optimized to the holotomography data, you may want to. It may work to other data format and other type of principal imaging the technique, but the accuracy may not be very high. So I believe for specific application of specific technique, you need to optimize. You need to develop the algorithm independently, I believe. But I think this is very intriguing questions.
Absolutely. Okay, perfect. So those are all of our questions at this point. I would like to again thank everyone for attending. If there's any other questions, please feel free to reach out to us via our website, or we will be at AIM next week. Feel free to come and talk to us. I want to thank Paul again for his time and presentation. And thank you, everyone. We look forward to coming to Berkeley next week.
Thank you.
Bye-bye.